In 2021, the Illinois General Assembly passed and the governor signed a law creating the Illinois Forensic Science Commission, which is helping guide the future of forensic science in Illinois. Members of the commission have decades of experience as scientists, lab directors, lawyers, doctors, academics, and community advocates who strive toward the common goal of making a positive difference on the criminal justice system in Illinois. The Commission, in collaboration with the Illinois State Police Forensic Sciences Command, the DuPage County Forensic Science Center, and the Northeastern Illinois Regional Crime Laboratory, developed this Fundamentals of Forensic Science video series as an educational resource for the public and forensic science stakeholders. Each video in the series will educate viewers on the science, technology, and methodologies underlying one of the core forensic science disciplines and serve as a building block for expanded forensic science education.
Seized drug analysis within the state of Illinois. A controlled substance is a chemical compound that is regulated by federal, state, or municipal statute. It may be controlled by name or chemical structure. State regulations can be found in the Illinois Controlled Substances Act. In Illinois, controlled substances are categorized by their accepted medical value and potential for dependence and abuse. This ranges from Schedule I substances, which pose the highest risk for abuse and have no accepted medicinal use, such as heroin and ecstasy, to Schedule V substances, which pose a significantly lower risk and have widely accepted medicinal uses, such as the nerve pain medication pregabalin. Other examples of controlled substances and their corresponding schedules are listed here. In Illinois, cannabis is regulated separately through various statutes, including the Cannabis Control Act, the Cannabis Regulation and Tax Act, and the Industrial Hemp Act. Seized drugs are submitted to a laboratory as evidence suspected to contain one or more controlled substances. Items may be submitted as plant materials, powders, tablets or capsules, liquids, mushroom material, edibles such as beverages, chips, or candy, or paraphernalia, which may include pipes or bongs, scales, or syringes. When a laboratory receives seized drug evidence, a forensic scientist trained in the analysis of controlled substances seeks to answer two questions. 1. What is the weight of the item submitted as evidence? And 2. Is a controlled substance present? Forensic scientists may also report the presence of emerging substances of interest for intelligence purposes. Evidence received by the laboratory is subject to evidence handling procedures. Each item receives a unique laboratory number for internal identification. All evidence is tracked digitally within a Laboratory Information Management System, or LIMS, and is marked appropriately to show the chain of custody. Forensic scientists wear proper personal protective equipment, or PPE, such as laboratory coats and disposable gloves. All evidence must be appropriately sealed upon submission and prior to any subsequent transfer. Proper seals include heat seals or evidence tape seals. After a case is assigned, the forensic scientist will confirm the contents of the evidence container, weigh the evidence, test for the presence of one or more controlled substances, repackage the evidence, and write a report, which may be submitted for technical review. The reports are sent to the submitting agency, and occasionally the forensic scientist may be called to testify in court. Weighing of the evidence is the first step during the analytical process, as individuals are charged and sentenced based on the weight of the item containing a controlled substance. When a substance is weighed, it is normally placed inside a weighing vessel. The weighing vessel pictured at the right is a disposable weigh dish that will be used for a single unit and then discarded. The balance is tiered or set to 0 with the weigh dish on the balance pan so that the weight of the vessel does not get included or reported with the weight of the evidence that was submitted. After weighing the evidence, the scientist begins the testing process. This process utilizes a combination of analytical techniques known collectively as an analytical scheme. The techniques available to a scientist are like tools in a toolbox, each providing additional clues as to what substances may be present in the sample. Scientists use these tools to meet or exceed minimum testing requirements and draw a conclusion as to the contents of the sample. The analytical techniques within the scientist's toolbox vary in selectivity. This means that some provide more specific information about a sample than others. In general, SEA's drug analytical techniques are broken down into 3 categories, ranging from least selective Category C techniques at the bottom of the pyramid to highly selective Category A techniques at the top of the pyramid. Category C testing techniques only provide general information about a sample. They may allow the scientist to narrow down the options that a substance could be, but they do not allow for the determination of a specific substance. Category B techniques provide enhanced selectivity over Category C techniques by assessing the chemical and physical properties of a sample. Category B and C techniques together are often referred to as preliminary or screening techniques. Highly selective testing techniques provide information that allow the scientist to identify a compound by providing information related to the molecular structure of a compound. This is why these techniques are often referred to as confirmatory tests. Some of the preliminary testing techniques available to forensic scientists include ultraviolet visible spectroscopy, chemical color tests, chromatographic techniques, and physical identification of legitimate pharmaceutical preparations. Confirmatory techniques in a scientist's toolbox, providing highly selective structural information, include mass spectrometry and infrared spectroscopy. Scientists perform multiple tests on each reported sample, often of varying degrees of selectivity. This is referred to as an orthogonal approach to testing, which means that scientists utilize testing techniques that provide different types of data. When different tests provide data that independently corroborate the same result, scientists can be more confident in their conclusions. Preliminary or screening techniques fall into categories B and C of the selectivity pyramid. The following introduces some of the different screening techniques contained within the scientist's analytical toolbox. Preliminary tests may be utilized to help determine the direction of an analysis, or they may be used to help support a conclusion. They are never used as the sole test for deriving a conclusion due to their lack of specificity. One limitation may include the inability of a certain chemical color test to differentiate between compounds that have similar molecular structures, such as the opiate compounds depicted on the left. Chemical color tests are used to indicate the possible presence of a drug or class of drug contained within a sample. To perform this test, chemicals are added to a very small amount of the seized drug sample. Scientists observe and document any color change that may occur to determine if a specific drug or class of drug may be present. Ultraviolet visible spectrophotometry measures how light waves within the ultraviolet and visible regions of the electromagnetic spectrum interact with chemical compounds. A small portion of the seized drug sample is dissolved in a suitable solvent and placed into the instrument. As light radiation is shown through the sample, the instrument measures how much of the energy is absorbed by the compound within the sample. This absorption is recorded in a graph, like the graphs pictured at the right. Forensic scientists compare the resultant graph to graphs produced by known substances to deduce what compound or compounds may be present in a sample. Seized drug samples typically present as mixtures. Therefore, a process called thin layer chromatography, or TLC, can be used to isolate any potential compound of interest. To perform this test, a small portion of submitted sample is dissolved in a suitable solvent and deposited onto a TLC plate. The solvent is then allowed to evaporate so only the dissolved compound remains. 5 different samples have been deposited on this plate, one unknown sample in the middle, and known substances spotted on either side. From left to right, these include ibuprofen, caffeine, the unknown sample submitted as evidence, acetaminophen, and acetyl salicylic acid. The plate is placed in a chamber and the solvent is allowed to wick up the plate, carrying different components at different rates, depending upon the structure of each component and how it interacts with the surface of the plate. If one spot contains multiple compounds, these two will be separated as the solvent moves up the plate. Once the solvent reaches the top of the plate, it is removed from the chamber and allowed to dry before the scientist visualizes the spots and assesses their migration. In this case, the unknown sample has separated into 2 separate and distinct spots. Comparing these to the known samples, the lower spot traveled the same distance as the caffeine, while the higher spot traveled the same distance as the acetaminophen. There is no indication that the unknown sample contains either ibuprofen or acetyl salicylic acid. Therefore, the analyst can assume that this unknown sample contains a mixture of caffeine and acetaminophen, and will pursue additional testing to confirm. Another type of separation technique employed by forensic scientists is gas chromatography. This technique first requires the unknown substance to be dissolved in a suitable solvent and then injected into a heated instrument known as a gas chromatograph, or GC, where the sample is instantly vaporized and a stream of gas pushes the sample through a thin coated tube on the inside of the instrument, known as a column. The software program produces a graph for the analyst to interpret. The separation of the mixture results in separate and distinct peaks as seen in the image at the left. The retention time of each peak is the time in minutes between injection and detection and is dependent upon the molecular structure of the compound and how it interacts with the inside of the tube. Like most preliminary testing techniques, this test can provide valuable information to the analyst, but it cannot exclusively identify a controlled substance. Confirmatory tests are tests that provide more selective information compared to preliminary tests. Specifically, these allow the analyst to identify a detected compound. One of the 2 main confirmatory tests used by forensic scientists is mass spectrometry. This technique requires a sample to be separated into its individual components before each component can be identified. Therefore, mass spectrometry is most often used in tandem with gas chromatography. These 2 techniques together are referred to as gas chromatography mass spectrometry, or GCMS. Once the different components of the mixture are separated by the GC, they independently enter the second part of this instrument, the mass spectrometer. Here, molecules are broken into pieces or fragments, which get filtered by size and electrical charge. Different compounds produce characteristic fragments in consistent ratios. Because GCMS is a 2-part instrument, we obtain a chromatograph in which the retention time of separate and distinct peaks can be determined, courtesy of the GC, as well as a spectrum of molecular fragments unique to a specific compound, courtesy of the mass spectrometer. Here are 3 different spectra. Cocaine to the left, which also shows the GC peak chromatogram at the top. Heroin to the top right, and methamphetamine on the bottom right. The patterns created by the fragments of these substances vary greatly, but the numbers and ratios of the cocaine fragments can be clearly distinguished from those of the heroin and the methamphetamine. When run under similar conditions, cocaine produces a spectrum that looks like the one pictured here. The same is true for heroin and methamphetamine. These spectra are then compared to known spectra, either from in-house runs or reputable literature references for confirmation. Another confirmatory testing technique is Fourier transform infrared spectroscopy, or FTIR for short. This instrument utilizes radiation in the infrared region of the electromagnetic spectrum. A laser passes through the sample and causes the molecular bonds within the molecules to vibrate. This instrument measures and records the vibrational energy of a compound and produces a different kind of spectrum than the GCMS. These peaks or bands correlate to specific groups of atoms in the molecule. An example of cocaine base, or crack, infrared spectrum is pictured here. Comparison of an unknown to a standard allows an analyst to identify what substance is present. Here is an example of the same cocaine base spectrum compared to a spectrum of methamphetamine hydrochloride. There are several remarkable differences between these two spectra, which are primarily due to the differences in their molecular structures and how their individual atoms interact with each other. All of the data collected during the analytical process is used to generate a conclusion, which is communicated to relevant parties by a laboratory report. In addition to case identifiers, lab reports contain several key pieces of information, including a description of the evidence as it was received by the laboratory, the weight of the evidence, the results of the analysis, and finally the limitations of the analytical scheme, if applicable. Accredited crime laboratories maintain a quality assurance program to ensure accuracy in testing and reporting. As part of this quality assurance program, instrumentation undergoes routine calibration and function checks to ensure results are accurate and consistent. Analysts follow standard operating procedures when handling and testing evidence, and case files may be subjected to technical review prior to releasing the report. These steps help to maintain the integrity of the evidence and the reported conclusions. Seized drug samples are items obtained from law enforcement agencies that are suspected to contain controlled substances. Forensic scientists follow evidence handling and processing procedures as they utilize preliminary and confirmatory testing techniques to identify a substance present in a sample. The analytical process is guided by quality control measures to ensure accuracy of the reported results. For more information on the Illinois Forensic Science Commission, please visit isp.illinois.gov/forensics/Forensic Science Commission or contact forensic.science.commission@illinois.gov.